Hey everybody. So today I'm going to walk you through this blog post that I wrote on reconstructing dinosaur genomes from bird DNA and skeletons. I think this is really fun. I got really excited about this. I hope that after watching, you feel like this could be possible. Okay, so here is the main figure that I created, and this really shows the technique that I'm describing here, right? And it's a pipeline for inferring dinosaur genomes from modern birds and reptiles. So it's three different parts to this pipeline. You train a neural network to predict bird and reptile skeletal anatomy directly from their genomes, right? So here you turn the DNA molecule of a raven into a number on the computer, and you also have to turn the skeleton of a raven into a number on the computer. So you're turning it into a vector. This is a concept called morphometry and morphometrics. And basically, that's what machine learning is really good at right now, going from one number to another number, and trying to understand the mapping of one number to another number. And it's a quantitative or mathematical mapping. So you have to start by going DNA to bones first, because that's how it works causally in the world. All organisms go from DNA to bones, not the other way around, right? So when you start off, you have to start probably training the machine learning system in that direction. But then once you have a system and you've trained it on a number of different species and pairs of genomes and skeletons, then you can invert the model. And that's pretty common in machine learning. Kind of turning the model inside out or flipping it over. And there's mathematical ways to do that. So now you're retraining the model to go from skeletons to DNA, right? So here you see a crocodile skeleton being shown to the machine. And now it's learning the mapping from skeletons to DNA. So it's not just, one raven and one crocodile here. You're showing it many, many, many different species. And it gets better and better at making these predictions. So at a certain point, let's say you held out chickens. So you finish training the model, and then you give the model a chicken skeleton. And the idea is that hopefully it would be able to reproduce something that is similar to chicken DNA. You'd be able to test and see how good your model is. And if it's not good enough, there's a lot of things you can do to make it better. Show it more data, throw in more compute, improve the algorithm. And then finally, after you've finished training at this second step, you have something that you can basically throw any skeleton into, and it'll come up with a plausible recreation of a genome. Now, all dinosaur, besides birds, all dinosaur DNA is gone. There are no dinosaur genomes left on Earth. The idea is that any DNA evidence from fossils pretty much completely disappears after 1 million years. And dinosaurs lived 65 million years ago, right? So we will never have their DNA. So this is just trying to reconstruct plausible genomes. It's hypothetical, but at the same time, they could be functional genomes, and they may look very, very similar to the original animal if brought through development. So here, number 3, you can see feed dinosaur skeletal data into the inverted model to generate plausible dinosaur DNA. And I think this would really work. So we don't have the DNA, but we do have the fossil skeletons. And there's an incredible amount of math, an incredible amount of data in those fossil skeletons alone, right? We see those skeletons and our imagination goes crazy. But a machine learning system would break those fossils down into so many different patterns, and compare. A system that's trained like this would be able to compare those patterns to everything else it's seen, in a wide variety of different archosaurs, birds, crocodilians, reptiles, and possibly other animals like amphibians or even mammals you might want to include in the training set. And I think we could get pretty close to functional genomes for dinosaurs. And that's what we need to recreate them, right? We would need a genome. And so this is basically this is what you would need to create a Jurassic Park. And I think that advanced AI will bring it to us. So in this blog post, I talk about what future AI should be able to do for dinosaur resurrection. And then I talk specifically about this pipeline here that I came up with a few months ago. So let me start off by reading the introduction. I've dreamed of witnessing real-life dinosaurs since childhood, but the fact remains that we may never get our hands on original dinosaur DNA. Unfortunately, the genetic traces, even those trapped in amber, have completely degraded tens of millions of years ago, a major disappointment for dino aficionados and Jurassic Park fans. But as I've been watching the advance of artificial intelligence in the last couple years, I've realized that AI creates new possibilities. Modern machine learning has an astounding capacity for prediction, for finding hidden connections, and for cross-referencing. Generative AI excels at constructing domain-relevant data structures when provided sufficient context and training examples. And agentic AI is being programmed to plan, organize, and automate complex work at scales outside the abilities of people. Now, in 2025, it's rather easy to imagine a superintelligent AI in the future tinkering until it brings back a guesswork-based, life-sized biological recreation of a Tyrannosaurus rex. So even if creating precise clones of long-extinct creatures is impossible, in the coming years it does seem possible for advanced AI agents to create surprisingly accurate guesswork reconstructions of dinosaur DNA. This entry will discuss the information available to a superintelligent AI to help it piece together the clues necessary to do this. It will also propose a 3-step pipeline using machine learning to generate a synthetic dinosaur genome. This pipeline leverages existing data to gather information about something we don't have, dinosaur DNA. Essentially, we use data from modern birds and reptiles where we have both the DNA and the bones to train a system to recognize how genes shape skeletons. Then, by giving this trained system a dinosaur skeleton, we can ask, what kind of genes would have built this? In essence, it uses paired genomic and skeletal data from extant birds and reptiles to extract latent genetic signals and learn structure-function relationships, which we then apply to fossilized dinosaur skeletons to infer their genomic makeup. So allow me to offer an analogy involving analogies. Remember those analogical reasoning questions from the SAT? They would state two pairs of related things and then ask you how they share a common relationship. Here's an example. Blueprint is to building as genome is to body plan. All these types of questions would share the pattern A is to B as C is to D. Well, the technique laid out here shares that analogical structure. If A, a bird skeleton, is to B, bird DNA, as C, dino skeleton, is to D, dino DNA, then what does D equal? Bird genomes are related to bird skeletons, both high-dimensional vectors, by a complex analogy, a mathematical function. The math behind that analogy is beyond human capability to derive, but is tractable for machine learning. That is because it's less conceptual and more computational. But once the machine has quantified that analogy, it can apply it to dinosaur fossils to reason about dinosaur DNA. This is because the way a genome produces a skeleton in birds is analogous to how a genome must have produced a skeleton in dinosaurs. So in the rest of this entry, I talk about how this would work. And here I talk about current AI systems that are looking at genes. And then here I start to talk more specifically about the pipeline that I'm introducing. And here's a somewhat more detailed figure. And then I talk about other information aside from just skeletons that could assist in the reconstruction. So we also have a lot of soft tissue fossils, remnants from some of these animals that lived tens of millions of years ago. And that would help because we can compare them to the soft tissues and other structures of living reptiles and birds in the same kind of way. And we may be able to think about the kind of soft tissue structures that dinosaurs could have had, specifically by looking at their bones and comparing them to the bones of reptiles and birds. We may be able to make certain inferences about some of these dinosaurs may have had frills or dewlaps or horns that don't fossilize. So birds and reptiles provide a wealth of genetic and anatomical data. Here I show various animals and animals we think of as dinosaurs. Of course, ichthyosaurs, plesiosaurs, and pterosaurs are not true dinosaurs. And I show their closest living relative in the years since they diverged. So this is helpful because it tells us that when looking at ichthyosaurs or plesiosaurs or pterosaurs for that matter, birds are not going to help as much as reptiles help for those reconstructions. And I think it's pretty fascinating that mosasaurs are very closely related to snakes. So another thing that I point out numerous times in this essay is that when we're trying to make estimations about dinosaur DNA, we're not extrapolating. We really are interpolating, right? Because we have living crocodiles and we have living birds on the other end. And dinosaurs would have been related to both. And in a sense, they would have been between birds and crocodiles, with reptiles on the left as well. So we have dinosaurs available to us today in the form of birds. And then we have relatives of the ancestors of dinosaurs and other reptiles, the first of which are crocodiles. So it's hard to extrapolate mathematically and statistically, but it's much easier to interpolate. And we would definitely be taking advantage of this. There's a lot of morphometric and phenotypic diversity in modern day reptiles. And we'd want to take advantage of this as well. You can see that in living fossils like the tuatara. So here we're talking about dino de-extinction. And as you can see, birds and reptiles do offer considerable relevant diversity. When you're trying to build a dinosaur, we would be building it from all kind of animals that we still have today. And I think that the diversity really is there to rebuild dinosaurs. I think we're looking at it right here. The biggest bird that we have DNA from, the giant moa, is currently extinct, but we do have their fossils and we do have their DNA. And they would probably be very helpful to a dinosaur reconstruction because they were so large, they give us a lot of details on how birds get big and the genetics of gigantism in birds. And then I talk about a lot of related techniques that you might be interested in reading about. And then here in this diagram, I talk about a machine learning method for predicting extinct facial features from skulls. So here we're not dealing with DNA at all. We're dealing with pictures. So this would be generative AI, like the kind that we use when we create pictures using DALL-E or an image generator online. So we can train an AI system using a skull encoder and a face generator, probably a diffusion model or something like that. We train the model on skulls and matched faces of living animals to teach it how skull shape influences facial features. Then we use the model to infer and generate imagery of facial characteristics of extinct animals from fossil skulls. So I think this would also work for a large variety of different prehistoric beasts. And I think it would work for humans too, right? So we train a neural network to learn the mappings and then predict how human and ape skull shape influences facial features. Then we use the model to infer facial characteristics of extinct human ancestors and other hominins from skulls, particularly fossil skulls. So we have some really cool human ancestors that are more than 1 million years old who will probably never have their DNA. But I think that we could see their faces for the most part, reconstructing them from the diversity that currently exists in primates, especially apes and humans, because I truly believe that a properly programmed AI system that's been exposed to all of that natural diversity would be able to use that diversity to recreate things that are lost. Here's Jack Horner's method, and of course this is very different from the method that I am espousing here. Basically, he wants to take an existing bird, like a chicken, and he wants to find out how some of its earlier dinosaur features have been inhibited genetically and basically disinhibit. He thinks that a lot of dinosaur traits, or the genes that code for certain dinosaur traits, do still exist inside of birds, but they've been masked or repressed. And there is a lot of good evidence showing that you can turn a beak into a snout by changing genes. You can turn a gape into teeth, wings into arms, and a pygostyle into a tail. So this would not be bringing back individual dinosaur species based on their skeletons. This would be taking a bird and trying to revert it back to a more dinosaur body type. And fascinatingly, this is an example of some scientists who did take, I believe it was a chicken, and suppress the beak, suppress the genes responsible for a beak, and they really did turn it into something much more like a dinosaur that doesn't have a beak at all. And they didn't let it grow to maturity for legal reasons, but I think that's pretty fascinating. So we ask what's next for de-extinction, and I believe that this process that I'm talking about here, that 3-step pipeline that we talked about above, is not just applicable to dinosaurs, but it's applicable to any animal that we have good fossils of and we have relatives of. We have their body types, their bodies, and their genomes. And I think it would be possible to bring back things from trilobites to Gigantopithecus to Australopithecines. Here, like I said, I think this would even be possible to use with plants, this technique, or something like it. I think that this pipeline is currently the best way to do this, to reconstruct a plausible genome. And And I think it will be used by AI systems in the future. So here I give a whole bunch of different extinct animals and talk about whether or not we do have their DNA or not. Here are some extinct hominins and other human ancestors. And as you can see, for most of them, we do not have DNA. But I think we could reconstruct that. And that would be much easier to reconstruct than the DNA of a dinosaur because they lived much more recently, right? Then I talk about some of the weaknesses of the approach and try to come to a conclusion. Yeah, guys, thanks for going through this with me. And let me go ahead and finish by reading the conclusion. One of the fondest memories I have is of reading Michael Crichton's Jurassic Park in 3rd grade, before the first movie came out, and marveling at the idea of extracting dino DNA from a mosquito trapped in amber. Today we know any DNA contained within those mosquitoes has completely decomposed, but it sure felt elegant at the time. Even without DNA recovery, AI promises a new kind of virtual paleogenetics, a way to infer and simulate the genomes and physiologies of extinct organisms using bioinformatics, sophisticated prediction, and comparative analysis. This is in part to another scientific concept that enraptured me as a child, and the original Jurassic Park novel got right, that birds are dinosaurs. Despite the major hurdles, the method outlined here could be a good starting point for several reasons. Creating the first genome-to-skeleton bidirectional models would produce incremental value at each step, advancing the understanding of genotype-phenotype interactions. Even without resurrection, these AI-generated dino genomes could allow synthetic cell lines for studying dinosaur biochemistry in vitro, organoid models, or simulations of growth curves, muscle structure, and thermoregulation. The present method would only produce a synthetic consensus dinosaur genome, not a true historical sequence. It would be educated guesswork, but firmly grounded in real data. It could even result in living and breathing organisms that look how we expect dinosaurs to look, although they may not function or act like the real thing. I believe that creating a Jurassic Park with cloned approximations will be possible when methods like those discussed here are in the hands of superintelligent AI agents. In fact, I wouldn't be surprised if this fantasy could be brought to life within our lifetimes. And that there at the end, it brings up a good point that I discussed earlier, that it'd be much easier to recreate their bodies than their brains. And the instincts of T-Rex would be very difficult to resurrect. And another issue that I talked about earlier, if you end up reading the article, is that even after you have the genome on a computer, and you have a plausible genome that could be possibly turned into, a real T-Rex or a real dinosaur, scientists currently don't have the ability to turn that genome, which would be a list of A, C's, T's, and G's, right, into a full molecule. They can do several hundred bases, I think, at a time, but not billions of bases. And then even if you were able to create the entire molecule and try to insert it inside of an egg, you're going to have a lot of problems because we don't have dinosaur eggs, and an ostrich egg is not the same as an egg produced by a mother Tyrannosaurus Rex. And there would be all kind of different genetic compatibility issues. But the main point of all this that I'm discussing is that artificial intelligence now in 2026 is advancing at an incredible rate. And I don't think it will be that long before a technique like this described here is very possible. And some of these techniques that are beyond humans right now, I think are going to be available to super intelligence and advanced robotics. And I'm excited about the possible resurrection of some of these creatures. And we have to remember, though, that these animals may not be resurrected fully as living, breathing, moving beings, but they may be brought to life more completely and more realistically in movies or in digital simulations or recreations. And I think that's exciting as well. So thank you guys for going through this with me. There's a lot of detail in this blog post that I didn't talk about here. You can find it at my blog, Observed Impulse, or by Googling this title right here. Thanks so much for your time.